I was watching a couple at a crowded airport terminal last week, and it really felt like I was observing two entirely different operating systems.
Wow, one of those moments.
Exactly. They're trying to process the same stream of data, but in completely different ways. The woman, she was just hyper alert. Her eyes were constantly flicking, from a crying child over there to the way a security guard shifted his stance.
She's doing high-stakes social navigation.
That's it. She's reading the room's emotional temperature before a single word is even spoken. But beside her, the man seems almost, unnervingly detached.
Yeah.
He wasn't oblivious. He was just filtering. His focus was locked on the exit routes, the luggage logistics. His face was this mask of defensive regulation. It just cut through all the social noise like a scalpel.
And we usually chalk that up to what? Female intuition or male stoicism.
Right, as if they're just personality quirks or, cultural habits we picked up. But a landmark study that just came out of Yale suggests a much deeper, more clinical reality.
And that's what we're digging into today.
Exactly. That divergence in how they process the world, it wasn't a choice. It was the result of a low-level patch that got installed in our genetic code millions of years ago.
And that patch has a name. Human Accelerated Regions, HARs.
HARs. These are the parts of our DNA that just evolved at breakneck speed after we branched off from chimpanzees. They're like evolutionary editors, rewriting the developmental script of the brain.
But here's the turn, right? The edits didn't create one single unified human mind.
No. Instead, they acted as a diverting valve, precision-tuning neural circuits in different directions based on these ancient trade-offs, things like parental investment and sexual selection.
It paints a startling picture. Our mind isn't this finished masterpiece of rationality. It's the site of a silent genetic arms race.
We love to think we're the authors of our own social behavior. But beneath the surface, our brains are still running this Stone Age blueprint, and negotiated compromise between maternal and paternal genes that are often in direct conflict.
Which really makes you wonder, in our modern lives, are we actually making the decisions?
Or are we just watching the functional echoes of an ancient tug-of-war play out in our own heads?
That is a dramatic and a very compelling starting point. It perfectly sets up the mission for this deep dive.
Right.
Because the core idea here is that our modern social behavior, the way we interact right now, might be driven by these conflicting ancient genetic blueprints.
So we're going to look at the genomic origins of that divergence, the blueprint that governs, everything from the size of our brain to how we anticipate danger.
That's the mission. We need to unpack the genomics behind this evolutionary acceleration, understand what those edits actually did to the developing human brain.
And analyze the steep costs.
Right.
The vulnerabilities.
The vulnerabilities, the diseases. And then finally connect those ancient regulatory changes to the strikingly divergent neurobiology we see today when people process social threat.
Okay, let's unpack this, starting with what exactly these human accelerated regions are and why they were initially so hard to study.
To really get a handle on a human accelerated region, you have to appreciate deep evolutionary time. The entire definition is built on contrast.
Contrast with what?
With the rest of the vertebrate genome. These are sequences of DNA that are just incredibly conserved across a vast number of species.
When you say highly conserved, you mean they just don't change?
They don't change. The exact sequence of nucleotides, the A's, C's, G's, and T's, has stayed virtually unchanged for, maybe 100 million years or more?
So we're talking about sequences that are older than the dinosaurs, sequences that mice, whales, and chimpanzees all share. Pretty much verbatim.
Exactly. They represent the stable core programming, the stuff that's absolutely necessary for basic vertebrate function. Their lack of change is itself a profound evolutionary signal.
Meaning that they perform some kind of indispensable, highly constrained function.
Precisely. Any change in those sequences for millions of years would likely have been detrimental. So it was selected against, the system worked, so evolution said, don't touch.
But then, along comes the human lineage.
Right. And in the 6 million years since we split from the chimpanzee ancestor, these segments suddenly, they acquire this unexpected, sometimes really dramatic number of nucleotide changes. And they do it rapidly, specifically in our Lyme.
Okay, so something that was frozen for 100 million years suddenly thaws out and starts evolving like crazy, but only in humans.
And that rapid divergence suggests that the extreme constraint they were under was either lifted or, and this is the far more exciting possibility, they were being actively modified to enable some fundamental human-specific trait.
This immediately flags them as the prime suspects for the genetic basis of what makes us human, especially our cognition.
They had to be. Where else would you look?
I remember when these were first discovered, around 2006. And the immediate challenge was that most of them weren't in the protein coding genes. They were in what people used to condescendingly call junk DNA.
Right, the junk DNA problem. It was a huge mystery.
If they weren't building proteins, how did researchers even begin to prove they were functional at all?
Well, that gap in understanding really defined a lot of genomic research for years. We could see this rapid evolutionary change happening. We knew it was important, but it was all in the non-coding regions of the genome.
The dark matter of the genome.
A perfect way to put it. The breakthrough came from major technological advances that allowed us to finally interpret the function of this regulatory DNA. We're talking about really sophisticated tools like epigenetic profiling.
Which is like mapping, where regulatory proteins physically bind to the DNA.
Exactly. And things like massively parallel reporter assays, where scientists can test thousands of DNA sequences for regulatory activity all at the same time. It's a brute force approach that became possible with modern sequencing.
So it's like we went from having a static printed map of the genome to having a dynamic real-time wiring diagram.
Precisely. And thanks to those tools, and then later confirmation using things like transgenic animal models, we now have really consistent confirmation. The vast majority of HARs function primarily as gene regulatory enhancers.
So there it is. They aren't the instruction manual for the components themselves.
No.
They're the switches,
Yeah.
the levers, the dimmer knobs that control when, where, and how much of those components get built. They're in charge of the production schedule.
That's the perfect analogy. Enhancers are regulatory elements that control the expression of nearby target genes. They don't change the protein sequence one bit, but they control its dosage and its timing in very specific cells.
And the critical finding, once researchers could map their activity, was their specific focus.
It was their focus. It was stunning. HARs are overwhelmingly enriched for activity in neurodevelopment datasets. They consistently correlate with being physically close to genes we already knew were involved in brain development.
So you have this rapid sequence change, and it's linked almost exclusively to the neural regulatory program.
That connection is. It's the most compelling evidence we have for the genetic basis of human cognition.
Okay, but if they were functional and locked down for millions of years in other species, and then we came along and rapidly edited them, how did researchers prove that the human version is actually functionally different from, say, the chimp version?
That required some really rigorous comparative functional genomics. It's one thing to see a difference in the code, it's another to prove it does something different.
So how do you test that?
They took the human HAR sequence and the chimpanzee ortholity, the exact corresponding sequence from the chimp genome, and they inserted them into what are called reporter assays, often inside living organisms like mouse or zebrafish embryos. The reporter gene is usually something that expresses a visible color, like a fluorescent protein. This lets the scientists literally see where and how strongly the enhancer sequence is driving gene expression.
A side-by-side comparison in a living system. And what did it reveal?
When they compared the human and chimpanzee sequences in these validated enhancers, the results were striking. 32% of them showed observable functional differences in expression.
A third of them were measurably different.
Yes. This wasn't just random genetic drift. This was tangible evidence of real functional evolutionary change in the regulatory potential of our DNA.
Let's talk about the classic example, HAR-5. How did a few nucleotide changes in this one regulatory region lead to something as profound as altering the size of our brain?
HAR-5 is one of the clearest examples we have of a HAR influencing a major anatomical trait. It functions as an enhancer for a gene called FSD8.
And FSD8 is?
It's a key component of the Wnt signaling pathway, which is a fundamental system for cell communication during development. The human-specific changes in HAR-5 were found to influence cell cycle acceleration in neural progenitor cells, or NPCs.
Wait, hang on. I thought we were going to say that human brain development is slower overall.
Yeah.
How does an acceleration of the cell cycle help with that?
That's where the detail is so crucial. The key is when this happens. It's what happens before the NPC commits to becoming a neuron. If you can accelerate the cell cycle during the proliferative phase, the phase where these progenitor cells are just multiplying to create a larger pool of cells, you rapidly increase the size of that initial pool.
I see. It's like a factory.
Yeah.
If you can churn out more workers quickly at the very beginning, you have a much larger workforce to begin the actual complex construction process later on.
Exactly. A larger progenitor pool means a larger final cerebral cortex. So HAR-5's evolution likely involved a fine-tuning that promoted this early rapid proliferation, setting the stage for a much larger brain structure to be built later.
That refines the picture so much. It's not just make brain bigger, it's a retiming of the foundational steps of brain building.
And we see complexity even within the HAR sequences themselves. It's not always just turning the dial up to 11. Researchers found evidence of what they call compensatory substitutions.
What does that mean?
Imagine that switch that regulates volume. One change in the DNA sequence might crank the volume way up, but another change immediately adjacent to it in that same HAR might pull it back down just a little bit.
So multiple changes are happening at once, some pushing and some pulling.
Right. The overall sequence has evolved very rapidly, but the net result isn't a total functional overhaul. It's often a stable, entirely new human function with highly precision-tuned expression levels. It's a series of rapid but stabilizing edits that converge on a new, very specific regulatory outcome that was required for our unique neurological demands.
That clarifies the basic mechanism.
Yeah.
So we have these highly conserved genetic regions that suddenly accelerate in humans, becoming these precision switches for neural development genes. Here's where it gets really interesting for me. If hearers are these evolutionary editors, how extensively did they actually rewrite the script for the human brain, and what key functions were augmented?
The term rewriting the script is absolutely apt, and the scale of this rewrite is what's truly staggering.
Okay.
Scientists conducted these comprehensive assessments of the regulatory activity of over 3,100 HRs. They tested them across a range of human neural cell types.
And the result?
The result was that nearly half of them, 49%, showed features consistent with reproducible, significant enhancer activity, specifically in neural cells and tissues.
Nearly 1 out of every 2 HARs is doing something substantial in our brains.
Yes. This isn't just localized tinkering in one corner of the brain. This is a systemic, widespread re-engineering of the entire neural operating system.
So was the evolutionary pressure just to make everything stronger? Just turn up all the neural function?
In many cases, yes. The pressure was definitely toward augmentation when they did comparative studies looking at differential enhancer activity.
Between the human and chimp versions?
Exactly. They found a very pronounced directionality. The human-specific sequence divergence at these HARs is predominantly associated with increased enhancer activity.
Comment more.
Approximately 68% of the elements that showed a differential activity exhibited greater activity for the human version than the chimpanzee version. This confirms a concerted evolutionary push to augment specific neural regulatory programs. It's what's fundamentally driving the human-specific neurodevelopmental pathway.
And this change is baked into the DNA itself, right? We're not talking about, environmental or external factors here. We're talking about the molecular instruction set being different from the get-go.
Absolutely. And that distinction is vital in genetics. We are dealing with cis-regulatory change.
Cis meaning on the same side.
Right, referring to variation in the DNA sequence itself. Researchers tested this by comparing the human and chimpanzee HARs in both human and mouse neural cell lines.
Why in mouse cells?
To see if the cellular environment made a difference. And they found that the differences in the cellular environment, what we call the transregulatory compartment, which includes all the external factors like transcription factors, it didn't significantly alter the overall enhancer activity of the HARs.
So the divergence is primarily driven by the changes in the DNA sequence itself. The blueprint changed, not just the contractor who was reading the blueprint.
That's a perfect summary.
Okay, so let's get back to this paradox we touched on earlier, the massive expansion of the human cerebral cortex. Our brains are huge, but the data suggests this expansion is achieved by slowing down corticogenesis. How does HAR acceleration lead to a neurological delay, and why is that delay so critical for our higher cognition?
This is a classic example of something called heterochrony.
An evolutionary change in the timing of developmental events.
Exactly. The expansion of our cerebral cortex, the part that gives us our higher order functions, is directly linked to a greatly prolonged period of development.
How prolonged are we talking?
In a mouse, that critical period of building the cortex takes about 7 days. In humans, it stretches out beyond 130 days.
1 week versus more than 4 months. That is an enormous difference in developmental time.
It is, and it reflects the sheer complexity of the structure we're building. The cortex isn't just bigger, it's laminated in a much more complex way. We have 6 distinct layers that require the precise, sequential generation of different types of neurons.
And to achieve that, You need a longer period where the neural progenitor cells, the NPCs, can just keep proliferating before they commit to differentiating into specific neurons.
Exactly right. You need to build up that pool of precursor cells.
And here's the paradox. To allow for this overall expansion of the developmental period, the cell cycle of the individual progenitor cells themselves has to slow down.
Precisely. If the NPCs divided too quickly, they would exhaust their stem cell potential prematurely. You'd end up with a smaller, less complex cortex.
I see.
The neural progenitor cell cycle is substantially lengthened in humans. It can be up to 8 times longer than in mice. This evolutionary delay, this slowing down, is the necessary prerequisite for achieving our level of complexity. It allows time for more cell divisions, yes, but also for complex internal and external signaling, which ensures the correct lamination of the cortex.
That delay has huge implications, not just biologically, but socially, right?
Absolutely.
A fetus and an infant that require over 130 days of this complex neurological development are going to be profoundly dependent for a much longer time. This feeds directly back into that initial scenario of increased parental investment and social trade-offs.
That is the crucial link. The biological demand for a slower, more complex brain structure necessitated a massive increase in parental, particularly maternal, investment. The genomic trade-off is crystal clear. Complexity costs time, and time costs dependency. This sets the stage for the divergent behavioral strategies we see later on.
So we need to find the molecular switch that caused this delay. This brings us to the PPP1R17 mechanism, which feels like the smoking gun here. Walk us through how HARs actively slow this cell cycle down.
The PPP1R17 gene was identified as a really strong candidate. It's a gene that underwent extensive rewiring in its expression pattern, specifically in the human cortex.
And its regulation is controlled by specific HARs.
By 2 specific ACE, HAR-263T5 and HAR-2636. Using some very sophisticated genomic mapping techniques, researchers confirmed a long-range chromatin contact between the PPP1R17 promoter and these 2 HARs in cultured neural cells.
So they proved the HARs were physically interacting with the gene, actively regulating it.
Yes, the HARs are the conductors, and PPP1R17 is the instrument they're playing.
Okay, so what tune is it playing? What does PP1R17 actually do?
PP1R17 functions as a repressor. It represses key protein phosphatase complexes, specifically PP1 and PP2A.
And those phosphatases are?
They are crucial for regulating the cell cycle. You can think of them as the accelerator pedal for cell division, particularly for controlling the G1 to S transition.
The G1 phase is that critical checkpoint where the cell decides whether to divide again or to exit the cycle and differentiate into a neuron.
That's the one. It's the point of no return.
Okay, let me try to simplify this chain for myself and for you listening. We have these phosphatases, PP1 and PP2A, that push the cell cycle forward, making it go fast.
Right.
PPP1R17 comes in and represses those phosphatases. Therefore, the HR-mediated augmentation of PPP1R17 is essentially turning up the volume on the brakes.
That is precisely right. The augmented HR-mediated regulation of PPP1R17 increases the braking pressure on the system. And by doing that, it substantially slows neuroprogenitor cell cycle progression.
And that provides the specific molecular mechanism for the evolutionary delay, the heterochrony, that allowed for the massive expansion and complex lamination of the human brain.
It's one of the most compelling pieces of evidence we have, linking a specific HR mutation directly to a defining human trait. It's a beautiful piece of molecular detective work.
That is a profound insight. This rewiring, slowing down the brain's fundamental development to build a better, larger model, is clearly the basis for all the complex social and cognitive traits that define us. But complexity always comes with a price.
Always.
And if the system is designed on such a fine-tuned precision delay, any disruption must lead to, well, to immediate catastrophic results.
That is exactly the next step in this whole story, the cost of acceleration. HARs built our complex human brain, but they also introduce unique vulnerabilities, which leads directly to how they're implicated in neurodevelopmental disorders.
When you look at the genes being regulated by these HARs, you realize they're incredibly high-stakes targets. Why are these specific regulatory regions so prone to introducing disease when they get disrupted?
Because the genes that HARs regulate are fundamentally dosage-sensitive and highly constrained.
Dosage sensitive, you mean the level of expression, the volume setting of the protein has to be exact. It's a Goldilocks problem.
It's a total Goldilocks problem. Too much or too little leads to system failure. So if the HAR is the volume knob and it's supposed to be set precisely at 7.5, even shifting it to 7.0 or 8.0 can cause a catastrophic malfunction.
How do researchers confirm this dosage sensitivity?
They looked at the haploinsufficiency scores, or HI scores, of these genes. Haploinsufficiency means that having only one functional copy of the gene instead of the usual two is enough to cause a disease.
So these are genes that cannot tolerate even a 50% drop in their expression level.
Exactly. And the genes that HARs regulate have elevated HI scores. They are regulating the master switches for neural development, for neuronal differentiation, for axonogenesis. Any disruption here reverberates through the entire cortical structure.
And that manifests as these severe phenotypes.
Yes, like abnormal brain morphology, schizophrenia, and very notably Autism Spectrum Disorder, or ASD.
The idea that mutations in what we used to call junk DNA could be responsible for something as complex as ASD is, it's just staggering. How strong is the evidence linking these HAR mutations to ASD risk?
The evidence is becoming highly compelling. And the focus isn't on major deletions, but on really subtle point mutations within the enhancers themselves.
Single letter changes in the code.
Exactly. One landmark study focused on consanguineous populations, where recessive mutations are more likely to manifest because a child might inherit the same rare mutation from both parents.
So you can see the effect of having two broken copies of the HAR.
Right. They sequenced the entire set of HARs in these families and found that rare biallelic point mutations on both chromosomes in neurally active HRs showed a significant excess in individuals with ASD compared to unaffected controls.
That's it. What's the estimated magnitude of this non-coding contribution? I mean, how much of ASD could this account for?
The data suggests that non-coding HAR mutations could contribute to ASD risk in as many as 5% of consanguineous ASD cases, which is a substantial number.
That is substantial. And what about in the general population?
Well, even in non-consanguineous populations, they looked at rare de novo copy number variations, CNVs. These are small duplications or deletions in the DNA sequence that happens spontaneously.
Okay.
They found that CNVs that encompassed HARs were enriched 6.5-fold in ASD probands versus their unaffected siblings.
A 6.5-fold enrichment for copy number variations involving these ancient evolutionary switches is a huge red flag. But there was another surprising finding in that data related to sex, wasn't there?
Yes, and this is absolutely critical for linking back to our opening scenario in the airport. When they were analyzing those rare de novo CNVs involving HRs, researchers found a notable 2.48 odds ratio excess in females with ASD.
Wait, say that again? ASD is generally more prevalent in males.
That's right. But this suggests that when the regulatory system built by HRs is structurally compromised via one of these CNVs, females show a disproportionately high risk. It emphasizes a heightened fragility in the HR-regulated developmental program that's specific to the female brain architecture.
Wow. So that points to a genetic compromise that's inherent in the female-specific regulatory program. A vulnerability that aligns perfectly with the ancient trade-offs we were discussing, the cost of increased complexity and parental investment.
It seems to fit.
Let's look at some specific examples of what happens when an HR gets mutated. How does a tiny change in a switch lead to a full-blown disease?
We can look at the PTBP2 gene. This is a brain-specific splicing regulator, and it's essential for neuronal differentiation. Its job is to make sure that the correct versions of proteins are built in neurons.
A quality control manager.
A very important one. Researchers identified a small 5-base pair insertion-deletion mutation in a HAR enhancer for PTDB2. This is minuscule, but the impact was dramatic.
What was the functional effect?
When they tested it in a neuronal state, that tiny mutation caused a 40% to 50% reduction in the enhancer's regulatory activity.
It cut its function in half.
Yes. And because PTBP2 is so highly dosage sensitive, this significant loss of regulation likely stalls or severely disrupts the necessary maturation of neurons. The resulting defects in neurogenesis are believed to underpin the severe intellectual disability and ASD seen in the people with this mutation.
It demonstrates that just disrupting the volume knob can be as bad as losing the entire gene.
In many cases, yes.
Give us another example of a foundational gene being compromised this way.
Let's consider GPC4. It's a gene that's essential for synapse regulation and neuronal connectivity. In 2 separate families with ASD and intellectual disability, 2 distinct homozygous mutations were identified within an HAR enhancer for GPC4.
And the result?
Both mutations resulted in a measurable reduction of regulatory activity somewhere between 20% and 25%. Given that GPC4 is involved in forming and maintaining the communication lines between neurons, losing even a quarter of its necessary expression level during development can cripple the ability of the brain to form complex circuits.
And this regulatory fragility It extends across the board of critical neurodevelopmental genes.
Absolutely. Other HAR-regulated genes that have been implicated include CUX1, which is crucial for forming synaptic spines and dendritic complexity.
The physical connections between brain cells.
Yes. Also, MEF2C, which is associated with severe intellectual disability and epilepsy. Mutations there caused a 30 to 50% loss of enhancer activity in progenitor cells. And CDKL5, which is associated with Rett syndrome and epilepsy, where mutations caused a 25% loss of activity.
It seems like the human-specific version of the HAR is so critically balanced that any deviation, any reversion toward an earlier or dysfunctional state immediately results in a disease that attacks our most uniquely human functions: complex cognition and social interaction.
That's a great way to put it. The conservation of the human-specific HAR version is critical. It's an incredibly high-gain, but also high-risk regulatory system. The evolutionary divergence that made us human also introduced these very unique vulnerabilities.
The genetic editors that gave us a bigger cortex simultaneously made the instructional manual incredibly fragile.
That's the trade-off.
So if that regulatory system is so delicately tuned and we're already seeing evidence of sex-specific genetic vulnerabilities in these non-coding regions, that 2.48 odds ratio excess in females with CNVs, how does this fragile, genetically fine-tuned brain translate to the dramatically divergent social behavior we saw in the airport? How do we finally link the genomics of HARs and the cost of developmental delay to these sex-specific strategies for managing threat and vigilance?
This is where we bridge that gap from the genome to the moment-to-moment experience of the world. And that divergence in threat processing is real, and it's measurable.
So how do they measure it?
Researchers needed a way to measure the brain's fundamental alertness level, sort of independent of any social context, and then see how that alertness level interacted with different kinds of social stressors.
And the fundamental measure they used for that was the error-related negativity, or ERN. Let's make sure we define that clearly for everyone listening.
The ERN is an electrophysiological measurement. You capture it using EE. It's a rapid negative voltage deflection that's generated in the anterior cingulate cortex, the ACC.
And it happens when?
It happens immediately after a person makes an error or perceives some kind of discrepancy. But crucially, while it helps monitor errors, the ERN has been validated as a very robust, non-social index of hypervigilance or reactivity to threat.
So a more negative ERN signifies a brain that is more intrinsically alert, more sensitive to unexpected negative events. It's the brain's general purpose early warning radar.
That's a great term for it, yes. The early warning radar.
So they established the sensitivity of this internal radar system, the ERN baseline, and then they put the subjects through a social stress test.
Exactly. They used something called the virtual school paradigm. It's an ecologically relevant fMRI experiment. Participants were told they were going to anticipate receiving feedback from purported peers who had established reputations.
Reputations for what?
There were nice peers, mean peers, which is predictable harm, or unpredictable peers, which represents uncertain harm. This allowed the researchers to map the neurobiological response to very specific types of social threat.
Okay, now based on the high rates of anxiety disorders we see in adult women, you might expect that the baseline neural radar, the ERN, would be higher in females to begin with, was it?
That's the logical assumption, but surprisingly, no. This was a crucial finding. The basic neural measure of non-social threat vigilance, the ERN, was similar across adolescent males and females.
So the sexes started with the same basic sensitivity to danger.
They did. The divergence only emerged when that ERN measure was combined with the type of perceived social threat they were anticipating. The ancient regulatory blueprint expresses itself differently depending on the specific social context.
Okay, let's break down the male pattern first. If a male subject had a highly vigilant baseline, so a more negative ERN, how did his brain process those social threats?
The male high vigilance circuit is tuned toward the detection of uncertainty. Males with a high non-social threat vigilance showed their strongest brain activation when they were anticipating feedback from the unpredictable peers.
The maybe danger.
The maybe danger, exactly. The unpredictable peer represents the highest degree of immediate, volatile risk. This aligns with an evolutionary strategy that would be focused on proactively neutralizing ambiguous or immediate physical threats.
And which brain regions were recruited for this proactive, uncertainty-focused strategy?
The heightened activation was seen in regions associated with detection, motor planning, and involuntary attention. Specifically, the left precentral gyrus and the left putamen.
Let's unpack those. The precentral gyrus is involved in preparing and executing actions. Right. It's linked to vigilance and motor response.
Correct. And the putamen, which is part of the striatum, is a key hub for evaluating context, for filtering sensory information, and for orchestrating fear conditioning in situations that are defined by uncertainty.
So the male high vigilance strategy is proactive and external. They prioritize intense monitoring of the source of ambiguity, perhaps preparing for a rapid involuntary attentional shift or even a physical response. They're filtering for the unknown quantity.
That's right. The pattern indicates a focus on proactively mapping and monitoring the external social environment for unpredictable dangers. And this could potentially lead to faster, more impulsive reactions before the threat is fully realized or evaluated. It's a system designed for immediate risk mitigation response to volatility.
Okay, now let's flip that and look at the female pattern. If the female baseline radar sensitivity was the same, how did highly vigilant females process these social threats?
The female pattern was characterized by internalizing and generalizing predictable harm. Females with high non-social threat vigilance showed their strongest brain activation when they were anticipating feedback from the mean peers.
The predictable threat.
The predictable threat. In this scenario, the threat is a certainty. You know the feedback is going to be negative. And this requires a different, more internal and more memory-driven strategy.
So which regions were engaged when anticipating this certain predictable harm?
The increase in activity was observed in the left amygdala and, crucially, the left anterior hippocampus. The amygdala's activation makes sense. It aligns with standard threat detection and fear response.
But the heightened activation in the left anterior hippocampus, that's the key functional divergence.
That's the key.
Why is the anterior hippocampus so important here?
The hippocampus is the seat of memory and context. And its heightened engagement here is consistent with the evaluation and generalization of that predictable threat across different contexts. The female system, when it's highly vigilant, is not focused on the ambiguous source of external risk, which was the unpredictable peer. It's focused on storing and processing the certain negative emotional outcomes from the mean peer.
That immediately links back to the high costs of complexity we discussed. If the human-specific female architecture is more fragile, as suggested by that CNV axis in Section 3, and the behavioral system emphasizes this deep evaluation and generalization of predictable harm, well, that's a perfect recipe for anxiety.
It's a very strong biological hypothesis. The mechanism of threat overgeneralization, which is taking a specific negative experience and applying that expectation to broader social contexts, is a known factor in the development of social anxiety symptoms.
And the heightened hippocampal activation suggests that's exactly what's happening.
It suggests that the high vigilance female strategy is geared toward minimizing long-term reproductive and social risk by deeply encoding relational threat. And this may help explain the increased prevalence of social anxiety symptoms in females despite them having similar non-social threat baselines.
So what does this all mean then? The foundational components, the general threat radar, are similar across the sexes. They likely evolved from the same initial ATR-driven changes that built the cortex in the first place. But then that system is channeled through these sex-specific regulatory pathways. This causes males to specialize in detecting the unpredictable danger using proactive externalized regions like the putamen.
Right.
And females to specialize in evaluating the predictable harm, utilizing internalizing memory integrating regions like the hippocampus.
It confirms that the divergence isn't just behavioral, it's deeply regulatory. And the different strategies are functional. They reflect these ancient trade-offs. The male pattern prioritizes a rapid response to volatility. The female pattern prioritizes deep encoding and generalization of relational harm,
which would be essential for maintaining a stable, safe environment for long-term infant development.
Exactly. It echoes the high parental investment costs imposed by the HAR-regulated slowing of the human cortex. It all comes full circle. Hashtag tag outro. We've really completed the entire circuit here. We've gone from these silent, non-coding stretches of our DNA through the molecular mechanisms that slowed down our brain's development to the genetic fragilities that arose from that very acceleration.
And finally, to the strikingly divergent strategies we employ today for reading social risk.
The knowledge takeaway is profound. Human cognition is built on evolutionary acceleration, but the regulatory code that resulted, the HARs, they dictated not only the size of our cortex, but also the compromises that led to the behavioral differences we see every single day.
It's compelling to realize that the mechanisms that gave us our large, complex cortex, that HAR-regulated slowing of cell division and the subsequent increase in dependency, are functionally inseparable from the ancient trade-offs that now dictate whether we focus on the uncertain external threat or the predictable internal vulnerability.
We are constantly running a dual operating system, and it's built on these historical genetic conflicts.
Yeah.
The specialization is so clear. One system geared toward proactive monitoring of volatility, which is often associated with male patterns.
And the other geared toward deep evaluation and internalization of predictable relational threats, often associated with female patterns. So the human mind, built on this HAR-accelerated complexity, it operates not as a single unified rational machine, but as a compilation of these specialized systems. And these systems, they were designed by ancient trade-offs. The need for hyper-alert social reading versus the efficiency of risk filtering, and they continue to run in conflict inside our heads. The evolutionary divergence is not a historical footnote. It is the active, genetically programmed mechanism underlying our most critical decisions about vigilance, trust, and threat today.